أسف على التأخير بالمحاضرة، كان أكو لبس بالتوقيت خاص بجدول المحاضرات. محاضرتنا لهذا اليوم عن جنس الباسيلس. The genus Bacillus includes large aerobic gram-positive rods occurring in chain. Most members of this genus are saprophytic organisms prevalent in soil, water, and air, and on vegetation such as Bacillus cereus and Bacillus subtilis. While Bacillus anthracis, which causes anthrax, is the principal pathogen of this genus. The typical cells are large gram-positive rods, have a square end, and are laid in long chain. Spore located in the center of the non-motile bacilli. Some of these bacilli or species of this genus are motile and some are non-motile. Colony of Bacillus anthracis, which is an important medical bacteria, are round and have a cut-glass appearance. يعني تكون المظهر مالتها اذا تخليها تحت الكاميرا مكبره بشكل قطع الزجاج المكسور. In transmitted light, hemolysis is uncommon in Bacillus anthracis, but common with the saprophytic bacilli due to its ability to produce a variety of enzymes and hemolyses that degrade RBC. Also, gelatin is liquefied. This bacteria can be utilized simple sources of nitrogen and carbon for energy and growth. The spores are resistant to environmental change, withstanding in dry heat and certain chemical disinfectants for moderate periods. Animal products contaminated with anthrax spore can be sterilized by autoclave. Now, the pathogenesis of this bacteria or Bacillus anthracis in a human, the infection or anthrax in general is primarily a disease of herbivores such as goats, sheep, cattle, and horses, and other animals such as rats are relatively resistant to this infection. Humans become infected incidentally by contact with infected animals or their products. In a human, the infection is usually acquired by the entry of spore through injured skin, such as in case of getting falls into the land and inhalation anthrax. Now, how the disease is occurred. The spores germinate in the tissue at the sites of entry and the growth of the vegetative organisms. Establish infection and colonize the sites of entry then the bacteria produce toxin or disseminate to other cells. Produce toxin results in formation of gelatinous oedema and congestion. Bacilli speed through or spread through lymphatics to the bloodstream, and they multiply freely in the blood and tissue shortly before and after death. Bacillus anthracis that does not produce a capsule is not virulent. So, capsule is considered one of the most important virulence factor of this bacteria. واحنا نعرف انه الوظيفه مالت الكبسول الاساسيه هي تعتبر انتي فاجوسيتيك activity. The polyglutamic acid capsule is anti-phagocytic. هنا راح نشوف انه the capsules of all bacteria are composed from polysaccharide, except for Bacillus anthracis, which composed from protein or amino acid, polyglutamic acid capsule. This is unique feature that distinguished Bacillus anthracis from other types of bacteria. يعني البكتيريا الوحيدة اللي الكبسول مالتها تكون protein in nature, while other are polysaccharide in nature. Anthrax toxin is made up of 3 proteins. Bacillus anthracis. احنا نعرف انه bacterial toxin is a bivalent, composed from 2 subunits, B called binding unit and A is called active unit. While the anthrax toxin is composed from 3 subunits or 3 proteins. The first is a protective antigen that's responsible for binding of toxin to the cell receptor. Then oedema factor and lethal factor. Protective antigen binds to specific cell receptors, and following autolytic activity, it forms a membrane channel that mediated entry of oedema factor and lethal factor into this. Oedema factor has an adenylate cyclase activity the adenylate cyclase with the protective antigen, it forms a toxin known as toxin. While lethal factor plus protective antigen form a lethal toxin, which is a major risk factor and cause of death in infected animal. As shown in this figure, encapsulated bacteria produce exotoxin. Exotoxin is composed from protective antigen, lethal factor, and oedema factor. This protective antigen binds to ATR receptor on the cell surface, which activates enzymatic activity of specific protein called furin and form a heptamer with this receptor. Then this receptor undergo endocytosis. After this, the oedema toxin is released, which lead to conversion of ATP to cyclic AMP by calmodulin activity, which lead to the oedema. While lethal toxin is lead to cell death through the activity of MAPEC, mitogen-associated protein kinase. In human, approximately 95% of cases are cutaneous anthrax, and 5% are inhalation. Gastrointestinal is very rare. Cutaneous anthrax generally occur on exposed surfaces of the arms or hand. A varietic papule develops in 1 to 7 days. The papule rapidly change into vesicle or small ring or vesicle that coalesces and necrotic ulcer develops. The lesion have a characteristic central black eschar, marked ordinal occurs, lymphadenitis and lymphadenopathy, and systemic signs and symptoms of fever, malaise, and headache may be active. After 7 to 10 days, the eschar is fully developed, eventually is dry, loosens, and separates. Healing is by granulation and leave an eschar. In as many as 20% of patients, cutaneous anthrax can lead to sepsis, systemic infection, including meningitis and death. While the incubation period of inhalation anthrax may be as long as 6 weeks. The early clinical manifestations are associated with marked hemorrhagic necrosis and edema of the mediastinum. Substernal pain may be prominent. Hemorrhagic pleural effusions can be developed and cough. Sepsis in these cases occurs, and they may be hematogenous separates to the gastrointestinal tract, causing bowel ulceration, or to the meninges, causing hemorrhagic meningitis, and the fatality rates and the inhalation anthrax is high. Now, lab diagnosis, including collection of specimen, which is maybe fluid, pus for lesions, blood, and sputum, or cerebrospinal fluid. Stain measure or immunofluorescence staining technique can be used to detect anthrax bacilli. This bacteria is cultured on blood agar plate. Organism produced non-hemolytic gray to white colonies with a rough texture and a ground glass appearance. Coma-shaped outgrowth, which is called medusa head growth or appearance. May project from this colony, ground stain, showed large gram-positive rods from median and from living tissue or infected tissue. Spore cannot be seen because of the organism or the bacteria is present in the vegetative form. طبعا معروف انه السبور يكون عندما تكون الظروف غير ملائمه لنمو البكتيريا undesirable for growth of the Bacillus entericis. ولكن في الانسجه المصابه the culture media the bacteria is converted from latent phase from spore to the vegetative form. فرير يعني انه احنا نشوف بالكلتشر انه وجود البكتيريا الحاويه على السبور. الموجود هو البكتيريا الحاويه على الفيجيتيتيف او اللي موجوده في مرحله الفيجيتيتيف فورم. Carbohydrate fermentation is not useful in semi-solid medium. Anthrax bacilli are always non-motile, whereas related non-pathogenic organisms such as Bacillus cereus exhibit motility by swarming. Virulent anthrax culture kill mice or guinea pigs upon intraperitoneal injection. An enzyme-linked immune assay has been developed to measure antibodies against adeno and lethal toxin. Molecular technique also can be used, such as using DNA hybridization, or most commonly using polymerase chain reaction technique for detection of this important bacterial pathogen. Treatments, including using of many antibiotics, are effective against this bacteria in human, like ciprofloxacin is recommended for treatment of anthrax. Penicillin G is in combination with gentamicin or streptomycin also can be used. And the setting of potential exposure to Bacillus anthracis as in an agent of biological warfare, prophylaxis with ciprofloxacin or doxycycline should be continued to 4 weeks. Some other gram-positive bacilli, such as Bacillus cereus, are resistant to penicillin by virtue of beta-lactamase production. Doxycycline, erythromycin, or ciprofloxacin may be effective alternative to this antibiotic. Now, epidemiology, prevention, and control. Soil contaminated with anthrax spores from the carcasses of dead animals should be managed and sterilized. These spores remain viable for decades. Control measures including disposal of the animal carcasses by burning or depuring in line pipes. Decontamination, usually by autoclaving of animal products, protective clothing and gloves for handling potentially infected materials, and active immunization of domestic animals with the live attenuated vaccine. Persons with high occupational risk should be immunized. Now, Bacillus cereus. This bacteria causes food poisoning and have two distinct forms: the emetic type associated with fried rice, and the diarrheal type associated with meat dishes and sauces. Bacillus cereus produce toxins that cause disease that is more an intoxication than a foodborne infection. The emetic form is manifested by nausea, vomiting, abdominal cramps, and occasional diarrhea, and is self-limiting. It begins 1 to 5 hours after ingestion of rice and occasionally past dishes. Bacillus cereus is a soil organism that commonly contaminated rice. When large amounts of rice are cooked and allowed to cool slowly, the bacillus spores germinate and the vegetative cells produce the toxin during log phase or during sporulation. Now, in the diarrheal form, as an incubation period of 1 to 24 hours and is manifested by profuse diarrhea with abdominal pain and cramps. Fever and vomiting are uncommon. The enterotoxin may be produced in the food or in the intestine. The presence of Bacillus cereus in a patient's stool is not sufficient to make a diagnosis of Bacillus cereus disease. This disease itself هتعرفون مشاكل الكهرباء الموجودة عندنا. Now, Bacillus cereus is an important cause of eye infections, including severe keratitis. Typically, the organism introduced into the eye by foreign bodies. Because of this organism presence in our environment in any site, and can be entered to the eye by trauma. Bacillus cereus has also been associated with localized infection and with systemic infections, including endocarditis, meningitis, osteomyelitis, and pneumonia. Now, thank you for your listens. Now, any question?